Today's talk will be about bioengineered EVs with fully native proteins, which Ciloa company is working on. Ciloa is the most experienced EV-based R&D powerhouse dedicated to the development of preventive and therapeutic medical innovation solutions using the cellular machinery involved in EV generation. Ciloa has the sole bioengineering technology to sort in vivo any proteins either free in EV lumen and or any membrane proteins embedded in EV membrane. More than 100 fully native membrane proteins have been sorted on EVs, like 30 GPCRs, kinase receptors, transporters, ion channels, and 20 viral envelope proteins. Its disruptive technology allows to first create therapeutic vectors with the potential to deliver specifically chosen cargoes to any type of cells, and secondly, develop virus-free and adjuvant-free vaccines against emerging viruses. In addition, thanks to its long experience in EV, Ciloa has implemented a robust and efficient process for the production of high quality. Today we are honored. He is a chef, executive officer and co-founder of Ciloa, and he's a PhD in molecular
So I think that Nazan is facing some problems with the internet connection. Maybe I can take over. Can you hear me well? Yeah.
And cellular biology, director of research at French National. Last academic position at.
Sorry, Nazanin, you had some problems with the internet connection, so I will just briefly introduce Robert Mamoun. So as Nazanin was saying, he is the chief executive officer of and co-founder of Ciloa, and he has a PhD in molecular and cellular biology from Bordeaux University. He's former director of research at INSERM, the French National Health and Medical Research Institute. His last academic position was as director of the UMS 3725 CNRS, Montpellier University, and which is one of the largest VSL3 facilities in Europe. He has more than 40 years in research in both France and the United States in molecular and cellular biology, virology, oncology, among other fields. He's co-inventor and inventor of patents at the origin of a disruptive technology of natural customizing exosomes, allowing the development of new generation of virus and adjuvant-free vaccines for therapeutic vectors. He co-founded in 2011 of the pioneer Ciloa company, which was winner of the Sanofi 2020 iTech Award. So thank you so much once again, Robert, for being here. And now I pass you the floor.
Thank you very much to invite me to present our company and our technology. So as you told, Ciloa is a company dedicated to the bioengineering of EVs and also the so-called exosomes. Next slide. Ciloa has 10 years of experience in extracellular vesicles R&D to generate new biologics. Just few information. You told that the company was founded in 2011 as a spin-off of Montpellier University and CNRS, and current CEO, it's myself, and Bernadette Rantin is the CSO and one of the owner of the patent. We are a small company in hand of only private investors and CNRS. The company is dedicated to the EV, the bioengineering of EVs using a proprietary and unique technology of in vivo EV bioengineering, founded on 2 international granted patent families, and we have since some years several others. We have fully freedom to operate with these patents, and thanks to the more than 10 years of experience in R&D, we have developed an EV bioproduction process that allow to obtain high yields of purified EV with a process that is fully scalable in the industry, and also with all the characterizations and analysis as in the state of the art in order to go to GMP grade of EVs that can be injected in humans. So the common, all of you know the common EV engineering technology, either chemical engineering or bioengineering. I will not talk about the chemical engineering because there are, in addition to the issues and the challenge of using EVs, add some other challenges like the safety and homogeneity of the EV. So I will talk only about biological engineering of the EVs, and all of you know probably that currently the methods of bioengineering proteins of the EVs to use already existing proteins of the EVs like CD63, CD81, clump proteins, etc., in order to graft either peptide, protein domain, or full protein at the surface of the EVs. But this lead to different issues. The first one is that you don't have a protein that can be grafted from scratch inside the EV membrane or inside the EV lumen. And second, and the most problematic issue is the fact that when you produce your protein of interest, you change the expression and you overexpress some protein, the regular proteins of the EVs, and with the risk of changing the fate in vivo. So our technology completely different and is founded on the proprietary small peptides that we have patented some years ago. And we use this pilot peptide to fuse this pilot peptide to the C-terminus, for instance, of a protein of interest. We construct a gene that encodes these fused proteins with this chimeric protein. And we have a chimeric gene that we introduce by cell transfection or cell transduction inside the cells. And the cell will do the job for us. We obtain the protein synthesis, and when the protein is fully synthesized with all post-translational modifications, it goes out, for instance, of the Golgi apparatus. And thanks to the pilot peptides, it is sorted by the EV machinery on the exosomes or EVs that are produced. So on the exosomes, you will customize the exosomes with any kind of protein embedded in membrane, free in the cytosol, coated under the membrane, any kind of localization, as you will saw after. So the first protein that we have direct sorted on EVs was a GPCR that we have produced in cells, and we have analyzed the EVs that are produced. So they are EVs, as you know, by NTA, you have a size of almost 100 or 120 nanometers of diameter. They contain all the markers, CD9, CD63, and CD81. But what concerning the protein that we have sorted on it, it was the GPCR-CXCR4. You can see on the Western blot that the protein is present in high amount, and the protein is not only present associated with the EV, but at the EV surface as the ELISA with antibodies directed against the external epitopes can show you here. So you have a strong, a high level of CXCR4 that are targeted inside the EV compared to the control. This is for one POC, but what are the topology? One question is the topology of the proteins for the CXCR4. You saw that the topology was correct because we can detect the protein at the by interaction of the antibodies with the external protein domains. But here we have another protein, which is the synaptotagmin protein, which is a synaptic protein that can be found in two topology. The first one is the protein with the N-terminus outside of the cell membrane, and the second one is the reverse topology in the synaptic vesicles. So you have in synaptic vesicles, you have the C-terminus at the surface and the N-terminus inside the synaptic vesicles. So by adding the pilot peptides on the C-terminus, we obtain the protein under the EVs, and we know that it is in this topology because by looking at the glycosylation, we can see that the glycosylation occur on the N-terminus, and this is one proof that it is in this topology. The second proof is that by using antibodies against either the N-terminus or the C-terminus, and you can see by immunocapture that only the antibody AG2B that recognize the N-terminus is able to capture the protein and the EV. So this indicates that the N-terminal domain is exposed at the EV surface. It is what we expected. It is identical to its topology at the cell membrane. It is the opposite at the topology on the synaptic vesicles. We can choose the topology as we want. In that case, we chose to put it in this topology. So can we also send different kind of different protein on the same EV? We have expressed the CXCR4 and the CD8 protein fused to the pilot peptides in the same cell, and we purify the EVs and we immunocapture the EVs either by the anti-CXCR4 or anti-CD8 antibodies. And you can see that when we capture the EVs with anti-CXCR4, we have the CXCR4 here, but we have also the CD8 proteins. And the reverse is true when we capture the EVs with the anti-CD88. We have also the CD8 inside the EVs, but we have also the CXCR4 inside. So this means that we have both proteins in the same EV. This is very important because it allows to target membrane proteins complexes out of cells on EVs, like ion channel complexes, kinase receptors, heterodimers of kinase receptor, homodimers, GPCR heterodimers, etc. So you can reconstitute any kind of complex of proteins, membrane complex of proteins out of cells on EVs. And this is the case with this ion channel, potassium ion channel KV1.2, and you can see other very important and interesting fact of this technology, that because this ion channel is present in 3 different forms inside the EVs, the 2 immature forms here are sequestered inside the cells, and only the mature form is present at the surface and of the cells where it is functional. So thanks to the EV machinery, only the functional mature form is sorted on the EVs. And this is the only technology in the world that allows to select only mature form from a mixture of different proteins inside the EVs. So this is a protein which is mature, which is sorted on the EVs. Does the protein is functional? Here you can see the functionality of the binding of its natural ligand, which is the dendrotoxin, and you can see that the binding is specific compared to all other control. This is very interesting because if the protein is functional, it means that the protein is present as a tetramer. So we have on the EVs reconstitute a functional tetramer of anion channels. More, we have other results that indicate that we can send also accessory proteins in the lumen. Now we have a technology that allows to reconstitute heterooctamers on EVs of an ion channel. So this was for the membrane proteins, which is a big challenge, but we have also other technology to load EVs with any cargo under the membrane in the lumen. So we have also this technology use the muricillate peptides like the SARC peptide and the pilot, always the pilot peptides. We put the protein of interest in between, in fusion in between both peptides. The first one targeted under the membrane, the second one targeted in the EVs. So here, this is the scheme, and this is what happened. If we use the nanoluciferase enzyme, you can see that it is well expressed in the cells, it is well sorted in the EVs, and you can see that it is there is the luminescence of the EVs, purified EVs also. So we have targeted associated to the EVs, this nanoluciferase. Does this protein is inside or outside, associated outside? It's a simple analysis using proteinase K said us that it is the case, the protein inside, because the proteins here, nanoluciferase and the pilot peptide, you can detect by both antibodies, it has the same effect. The proteinase K has the same effect as on ALIX. So I mean that when the EVs are intact, the proteins are protected from the proteinase K, and this is not the case of the CD81 protein. As you know, it is at the membrane. And of course, when you disrupt the membrane using Triton X100, you have accessibility of all the proteins, and all are digested. So the proteins, it means that the proteins is protected inside the EVs. And of course, we obtain the same with luminescence. You can see that compared to the positive control, in the presence of proteinase K, the luminescence is still there. This is for the proteins that is sticked under the membrane, bound under the membrane, but can we obtain free proteins like nanoluciferase at the surface? So we have developed another technology based always on this technology of proteins that is in between the SARC peptide and pilot peptides. And we call this protein carrier proteins. And these carrier proteins has the function to interact with interacting carrier protein interacting peptides. So this peptide is fused to a cargo. And this cargo can be, is sticked and interact tightly with the carrier proteins, normally, theoretically. And if you add a disrupting agent of our choice, we can disrupt and free these proteins. So next slide, you can see that this protein, nanoluciferase, via this interaction is targeted inside the EV here. Exactly as ALIX, and exactly as ALIX, it is resistant to the proteinase K. And exactly as ALIX, if you destroy the membrane using detergent, you have the proteinase K that can hydrolyze. And this is the same also for the luminescence activity. Compared to the positive control, all the luminescence is still conserved even in the presence of PK. So the protein is targeted to the EVs. It is inside the EVs. We can liberate free the proteins by adding a disrupting agent? Here are the EVs with ALIX, with the protein pilot peptide, which is this one, nanoluc, which is the other one. And if you add the agent, the disrupting agent, you can see that you the protein is not at all inside, or very little low level are present inside the EVs. So we are able to disrupt this interaction, and we are able to free this nanoluciferase inside the EVs. And of course, there is a control here. The luminescence is still there. And when you add the disrupting agent, you have very low level inside the EVs. So we have now a full technology that allow us to target at the membrane of EVs any kind of membrane proteins, even the most complex one, even with several transmembrane domain, even if they are trimer, tetramers, etc. We have also technology that allow us to target in the lumen of the EV a protein bound under the EV membrane or free in the cytosol. This is a really robust technology because we have more than 100 proteins that are targeted in the EVs, of which 80 membrane proteins like GPCR, for instance, or ion channel transporters, etc. So what can be the usage of this technology? What can be the utility of this technology? First of all, it's a very interesting R&D tool to study ligand membrane proteins interactions. As you can see here, if you have a chip of biosensor chip like BioCore chip, you can add and load the exosomes with the protein of interest at the surface. And if you add a ligand, you will obtain a shift of a laser beam that give you a signal that you can register. And by using this signal, you can determine all the K-on, K-G, etc. of the interaction of an antibodies, for instance, with the CXCR4 protein, for instance. So you can calculate the number of CXCR4 present at the surface. So here you see that you have 80 membrane CXCR4 on the surface of the EVs. So this is the first application, and even you can see different interaction differing from the antibodies. These antibodies recognize any kind of CXCR4, and these antibodies recognize only epitope of the CXCR4, which is present when the CXCR4 is in interaction with a small g protein in the lumen of the EVs. So you can see that almost half of the CXCR4 are in interaction with the small g protein. More, you can see also, you can look at the interaction with the chemokine, the natural endogenous ligand of the CXCR4, which is the SDF1 alpha. And you can see that when you add the chemokine, you have an increase in the signal, and an increase which is specific, because if you add a specific inhibitor, you don't have this increase. And you have also several information on the interaction parameters, and also another additional information which is given by the number of SDF that are interacting compared to the number of CXCR4. And by this calculus, you can have a simple calculus depending of the size of each protein, antibodies or SDF. You can obtain the information that all the SDF bind to all CXCR4 sites. So 100% of CXCR4 on EVs are functional for binding all the chemokines. So the proteins at the surface of EVs are fully functional, exactly as the protein, the former ion channel, potassium ion channel. So another interesting results is the use of this tool, SPR tool, to decipher interaction of different ligands. First of all, we have the chip, the biosensor chip of the BioCore with antibodies against synaptotagmin, the former that I discussed above before. So the exosomes the EVs has the synaptotagmin at the surface. They are captured by the antibodies, and you have a signal. The more EVs you add, the more signal you have. Next, you add a ganglioside, which is necessary for interaction of the synaptotagmin with the botulinum toxin. So you add the ganglioside, and you have an additional signal. Now you will add also the toxin. And now you have, depending on the amount of toxin you add, you have more and more signal. And now you have this kind of sandwiches with the antibodies, the EVs, the protein, the synaptotagmin, the ganglioside, and also the toxin. And by this way, you can decipher all the interaction parameters of each of these interactors. And now you can obtain the KD of this interaction. And this is really similar to the one and in line with that of the toxin on neurons cells. This one of the application, you see the power of the technology. Now, we are interested also to look at other applications and, for instance, to develop new therapeutic antibodies against challenging targets that are, for instance, GPCRs. First of all, this is a GPCR embedded in the membrane. You can see that only the external epitopes are accessible to antibodies, and this is the case by ELISA. You can see that you have antibodies against the GPCRs that recognize very well the EVs compared to EVs without the GPCR, though there are only the antibodies accessible. And this is very important that you don't recognize epitopes internal of the membrane or internal epitopes in the lumen. Only these epitopes, and there are the epitopes that are important to develop therapeutic antibodies. Now, these GPCR, for instance, are very well known as very bad immunogens. But to develop therapeutic antibodies, you must develop antibodies, first of all, and you must, of course, have a good immunogens. Some GPCR are known as good immunogens, so are medium immunogens, and some, like GPCR1, are known as non-immunogenic at all. Using all these GPCRs at the surface of the EVs, we have immunized mice, and we have quantified by ELISA the recognition of GPCR-EVs by the sera of these mice. And you can see that, of course, we have a recognition of these GPCRs, but even the GPCR1, which is well known as non-immunogenic at all, you can trigger and develop antibodies against this. And these antibodies are really specific of the GPCR because it's recognized by FACS. They recognize only GPCR cells and not the control cells. 100 times more, the signals is 100 times higher. So this is a very GPCR or challenging proteins at the surface of EVs has a real potential to develop antibodies and also for therapeutic vaccination. And this summarizes all these process. You know that to develop therapeutic antibodies against membrane antigens like GPCR, there are several bottlenecks. The first one is to have a perfect native membrane antigen, because if you don't have a perfect native antigens, you don't have a perfect antibodies. You need to immunize mice, for instance. You need to screen monoclonal hits, and also you need to characterize all the interaction parameters very easily. Thanks to the EVs, we are able to unlock all these bottlenecks. And you saw the results that you obtained. And so we are able now to develop antibodies against very challenging membrane proteins. This is for the first application. The second application is the development of EV-based vaccines. It is very simple to understand that if you have EVs and you add membrane protein, the envelope protein of virus, you can obtain something like that mimics the virus because it has the same size, it has the same shape. And so immunologically, you have something very potent because you have the perfect antigen, and more you can have the in certain conditions, you can have the immunostimulatory power of the EVs. So we are, for instance, here we have the example of the SARS-CoV-2 spike protein that we are able to target the protein, the S1 and the S2 protein at the surface of the EVs. The proteins are glycosylated, and they are present also, you can see in non-reducing Western blot, they are present as a trimer. So the SARS-CoV-2 spike protein on EVs are mature because they are cleaved in the S1 and S2 protein. They are glycosylated, and they are still associated in S1 and S2 trimer exactly, that fits exactly, mimics exactly what happens on the original virus. So after that, we have developed also a protocol to immunize mice. So with this chimeric DNA, with the protein of interest and the pilot peptides, we inject nucleic acid, DNA, for instance, in the mice 2 times in order to produce and to load with the autologous exosomes with the proteins of the spike proteins. So you obtain in situ, on autologous exosomes, something that mimics perfectly the virus. And after that, you proceed to a boost with the proteins on exosome that we have produced in cell culture. But this way, we can obtain any kind of a candidate vaccine that mimic HIV vaccines, Zika vaccines, chikungunya vaccines, or SARS-CoV-2 vaccine. And I will give you some results on the SARS-CoV-2 vaccine. Here you can see that after immunization of mice, you obtain neutralizing antibodies, and the level of neutralizing antibodies is higher than what was already published by competitors. You can obtain also very good immune response directed against either the S1 protein and the S2 protein. And this means that you will have high protection, and you expect a high protection and wide protection potential of this vaccine. It is a wide protection indeed, because if you compare the neutralizing antibodies obtained using only the spike targeted to the membrane, for instance, like a commercial vaccine that you all people knows, compared to the same spike protein with the pilot peptide in order to target the protein to the EVs, you have this kind of neutralizing immune response against the 4 different strains, SARS-CoV-2 strains, alpha, beta, gamma, and delta, from UK, Indian, Brazilian, and South African. And you can see that for the spike alone, you have a good immune response, neutralizing immune response, but very low against other strains. But this is not the case when you target the spike under the EVs. You have at least 3 times more titer and even 10 times more titer. And this means that you trigger more neutralizing antibodies. And what is very interesting is that the lowest level that you obtain is at the same range at the highest with the spike alone. So this means that even with an Indian, if you develop antibody vaccine with a UK strain, you will obtain something that will neutralize an Indian or Delta strain. So it can be considered as a pan-SARS-CoV-2 vaccine. It's really a proof of the power of this technology. Of course, we have a pipeline of several viruses, for which we have developed several antibodies. We have also the proof that they are neutralizing, and we have T-cell response and even protection with this kind of vaccines. Now we are moving to, we have also, of course, in the pipeline, more than 15 viral envelope proteins to generate vaccines against human and animal pathologies. Now we are moving to the generation of potentially therapeutic EVs that target with a targeting potential. So of course, if you have a fluorescent exosomes here with the ligand proteins at the surface, you choose the ligand proteins in order that it will fit to the receptor at the cell membrane, at the membrane of the cell that you want to target. So you have these EVs, fluorescent with a ligand. Of course, it will be captured by the cells and it will deliver its signal, its fluorescence inside the cells. So what happens if you do the same with the different ligands? Here, it's the neuronal cells on which we have put EVs with a ligand 1, specific on neurons or no ligands. And you can see by microscope that you have very well a good capture, high level of capture that you can see here in this high histogram. If you have other ligand specific or of another type of cells, a tumor cell, for instance, you have this kind of results. First of all, if you have the receptor coated on the plates, at 96 well plates, you can add different amount of EVs. So you have a perfect curve of dose response and capture response of EVs. And on the cells, you can see that there is a specific capture of the EVs by the tumor cells. So this is the proof that it works, at least in vitro. So now when you have different EVs with different potential for preventive, as a vaccines or as a therapeutic EVs, it is, and you need to have a good way to produce the EVs at very high amount, and also qualified EVs in order that you can inject them in men or in animals in a safe manner. So we have, thanks to our expertise and our long experience in EVs, we have developed all a process to produce and purify the EVs, and we have adapted this process from the T-flask and cell factory to bioreactor. And you can see that in bioreactor, the cells grow very well. They have exactly the change in metabolites as we expect. And we know that when we harvest at day 4, day 5, or day 6, we have always the same level of EVs that are produced per cells. So the EVs is regular, and it's possible with cells in a bioreactor. So the EV production in a fixed bed bioreactors reproduce perfectly the EV production in T-flask or in cell factory. So we are able now to go in by increasing the size of bioreactor till having billions and billions of cells and millions of billions of EVs. Now we have in hand several grades of EV purification, because from the bioreactor we can go to a typical TFF equipment that allow us to concentrate and diafiltrate the EVs in order to have a concentrate and EV enriched fractions. We can semi-purify the EVs by exclusion chromatography and also obtain pure EVs by ion exchange chromatography. So this is for the production and purification. There is also, you need also, when you go in clinical phase, you need also to have dedicated quality controls in addition to all the quality controls necessary for all biotherapeutics. You need to have dedicated control, like you need to know the size, the homogeneity, the number. So you have all this information here. The presence of surface marker, you have this information either here or by Western blot, and you need to have markers of lumen, like helix, absence of different other markers that are contaminants, and you can find that can be originates from apoptotic vesicle, for instance, or blebs from cells. So we have all in hand, all these quality controls we have in hand. You need also to have control for the protein you have added at the surface or inside the EVs. So by ELISA, you can have it. You can have it by Bioacor. You can have it also by other instruments. You can have also the information on the presence of the cargo here by Western blot. And even you can have the information on some EVs, a population of, for instance, CD81 positive EVs. You know that, for instance, you have 40% of this population with the cargo of interest. This is very important. And we have a lot of information, and we know that almost in between 40 and 60% of the EVs that we produce contain the cargo either at the membrane or in the lumen. So I think there is also another thing that you must know before going to in the clinical is the stability of the EVs. And we have a study on the stability during more than 2.5 years with EVs stored at 4 degrees, -80, -20, -80, or lyophilized. And you can see that they are quite stable. You don't lose more than 30%. And the EVs, after 2.5 years, you have even the same, almost the same amount and the same quality of the EVs. So the EVs that we produce are remarkably stable in all tested conditions. And even with the CRCR4, it was also stable and present on these EVs after 2.5 years. So just to say that we have the full production process from adherent cells, with upstream process, downstream process, all the controls, and even storage conditions in order to go to do clinical assays in animals or in humans. So Ciloa has, just a summary, Ciloa has an advanced platform to develop new biotherapeutic with a bio-engineered EV production, with cell engineering, cell culture, purification, quality control, and storage. We have now the solution to produce pure bio-engineered EVs for clinical assay. I think that's all. And this is our team. Thank you. Thank you very much.
Thank you so much, Dr. Mamoun. It was great presentation and interesting, especially it was about EVs production in working industry. Okay, I'm going to ask if anyone have question. You can turn on your cam or hands up, or you can ask in group chat. Okay.
I think that there is at least one question. What is the pilot peptide? The pilot peptide, the pilot peptide is a peptide of almost 45 amino acids that we have when I was academic lab. I decipher all amino acids, and we know that there are 2 tetrapeptides that are proline, serine, PSAP, yes, which is known to interact with the escort machinery, and YXXL. These 2 peptides are very important to the functionality of the pilot peptides. And we have a paper in, I don't remember, 10 years ago, and a little bit more than 10 years ago, that described these peptides. So the important characteristics are these two tetrapeptides that interact with the escort machinery in order to hijack the escort machinery. And the very interesting thing is the fact that as the escort machinery is very conserved in all mammalians and even in other animals, the pilot peptides can play its role in any kind of cells, even mesenchymal stem cells, even mouse cells, human cells, rabbit cells, any kind of mammalian cells. So the next question, it was previously shown that mystery malmutation. No, yes and no. Yes, this is a very interesting question because there are some people that described and published that the SAR peptides in 2011, I think, help to target protein inside the EVs. Our discovery of the role of the SAR peptide with the pilot peptides has 12. It's before the 2010, so it's before the publications. So it is well known. Of course it is important, but it is not always sufficient. From time to time, depending on the protein, you can target the protein inside the lumen of the EVs, but it doesn't work always. You need to have the pilot peptides in order to improve and to have 90% or 100% of chance to obtain the targeting inside the lumen. This is important. We have compared this, and from time to time it doesn't work, but nobody's knows because all proteins have their own proprietary and properties, and they go in the pathway they want. And so you need to have a real balance that push your protein inside the EVs. Sometimes it doesn't work. And there is a last question, I think. Yeah. Which cell do you use for the bioreactor? How does the bioreactor works? Okay, which cells? You can use probably any kind of cells because it's not these bioreactors is not dedicated to the cell that we use. That are HEC cells, HEC 293 cells. They are work in the bioreactor. How does the bioreactor works? You have it's a fixed bed bioreactor because we are fully convinced that the cells must be in their best environment, and the cells that we use need to be adherent. And if you have some cells in culture, in suspension, you have much more debris, you have much more blebs, and you have much more apoptotic vesicles. And at the end, when you purify your EVs, you have blebs, apoptotic vesicles that you cannot separate from the real EVs. So it's very important that the cells are in very good conditions. It's the reason why we use adherent cells and we use fixed bed bioreactors. What was the biggest challenge you faced when started the therapeutic EV company? Oh, it was a real challenge because during 4 years nobody's thought that there are future for the EVs. Nobody knows what are the EVs, and there are more and more people that know what are the EVs since 4 or 5 years. But 10 years ago, we are very, very few that believe in the EVs. So it was really hard. It was really hard because we are in Europe. It's much more difficult to have money in Europe than in USA, for instance. And it was difficult because we start from scratch. At the beginning, we didn't know if the technology works, what is the range of the working, what kind of protein we can target, what are the applications that we can look for. So we tried several things. So of course, we accumulate experience. But it was really a challenge. It was really hard. Now it's more easier because people know the potentials of the EVs and they believe in it. So, and we have now several, as you saw, so we have several proof that it will work, at least for some application, probably for not all, but at least for some application. There are now surface protein presenter available for EVs. What criteria are vital between them? So there are no, what you mean by several surface protein presenters. As I told you in the beginning of the talk, more or less, they use either chemical solution, and I hope that they will be able to go in clinical phase, clinical with this kind of technology, but I don't believe this. But for the other that bioengineer the EVs, they always use proteins that are at the surface of EVs. And when you never go from scratch, you never have a protein that never go on the EVs that you can target on the EVs without our technology. And more you know, and there are some publication on this. If you produce EVs that overexpress CD63, for instance, tetraspanin, you have another fate in vivo. So you will change the fate of the EVs, not due to the protein that you add, but to the protein that you the regular protein of the EVs that you overexpress. I don't know this is a good solution. With our solution, we have something that is more evident to use and to go in clinical with this and to have in vivo results. What is the product you are most proud of? At the beginning, I am a virologist, so the first thing I thought is to develop vaccines, of course. And the first protein that I present on the EVs was the hemagglutinin of influenza. And I will say, oh, fantastic, I will be able to develop any kind of vaccines. And now we know was not so evident. And there was several issues that we overcome. So now we are able to obtain this kind of results with the coronavirus. You see that we are above the other commercial vaccines, and even you can make a synergy between the mRNA vaccine and the EVs. What is the program? What could be your advice for young researchers that want to move for EV companies? The advice, it's complicated. To understand that is not so evident. You have thousands of papers that indicate, that reveal that there are huge potential, but be very careful. You have some results that works in the lab, but this is probably, and in our opinions, is not the case when you go in the real life. When you want to have a robust technology, this is a jump, a logarithm above. It is not obvious to transfer some results that you have in the lab in a company. So you must think about this. What is the robustness of your technology and how you can move it in order to have a safe product that you will be able to inject in men at the end. Do you know what is the percentage in your prep that carry? Yes, we know. We know, for instance, that now, at the beginning, we know that we have 80 molecules of CXR4 per EV. Now we have the information because we can know precisely EVs per EVs, what are the proteins that are at the surface. And we know that almost 40 to 60% of the total EV that we produce contains the protein that we want. So we can say that for CXR4, for instance, if we obtain 80 molecules per mean EV, it means in reality that we have 160 molecules per EV that are modified. And there are also other EVs that are not modified, but as you know, there are several pathways to produce EVs, and we are concerned only by some. Did you use CRISPR-Cas for engineering? Or if not, why didn't you use? We didn't use it, but we have all in hand to do it. But when you are a small company, you must focus. So as you saw, we have plenty of potential applications, and we try to have proof of concept in different applications, and we try to develop the less challenging one, because CRISPR-Cas9 is one application. If you are able to put any protein inside the EVs, you will be able to put the CRISPR-Cas9. I can bet this, but I'm not sure because I didn't do it. But you know that with EVs, you have several challenges before going in a therapeutic, in real therapeutic. The first challenge is the bioengineering. Second is the production, purification, characterization. The third one, to have a safe product. The third one, or another one, is the clearance in the body. And you know that the EVs are rapidly clearing. The next one is the targeting in vivo, which is different from the targeting in vitro. The next one is the delivery in the cell. Another one is the functionality of the protein. So you have several steps before going to the star. You look at the stars, but go first to the scale and step by step climbing, not going directly to the star. Of course, you will be able to do a nature or science, but you will not obtain something robust that you will be able to use. My method is not better or worse than the CRISPR-Cas9 because it's complementary. If you want to sort CRISPR-Cas9 robustly, always in the EVs, I think that our method will work pretty well. Thanks. Thank you, everybody.
If there is any other question. Okay. Thank you again. It was really an interesting session. I learned a lot from you. If you don't have anything to add, I think we can finish this session.
Thank you. Only bon courage for all.
Thank you so much for the presentation. It was really amazing.
Thank you very much. Bye-bye.
Goodbye, everyone. See you next time.
Thank you.